Biosynthesis of insulin. Insulin is a hormone produced by the pancreas, which helps to regulate the amount of glucose in the blood by aiding in its cellular uptake. Glucose is the main sugar found in the blood, and it is the body's primary source of energy. In pancreatic beta cells, insulin biosynthesis is initiated in the cytosol, as insulin mRNA is translated into pre-proinsulin. Pre-proinsulin is a single-chain molecule made up of a signal peptide, insulin B chain, C peptide, and an insulin A chain. Newly synthesized pre-proinsulin is brought across the endoplasmic reticulum membrane by the signal peptide. The signal peptide is then removed by a signal peptidase, forming proinsulin. Within the oxidizing environment of the endoplasmic reticulum lumen, the proinsulin rapidly folds. This folding forms 3 disulfide bonds, including 2 interchain disulfide bonds, B7 to A7 and B19 to A20, as well as 1 intrachain disulfide bond, A6 to A11. The folded proinsulin dimerizes and exits the endoplasmic reticulum. It is delivered to the Golgi apparatus, where it is packaged into secretory granules located near the cell membrane. During the maturation stage of the granules, prohormone convertase 1/3, prohormone convertase 2, and carboxypeptidase E cleave the C peptide from proinsulin to produce mature insulin and a free C peptide. The insulin and C peptides are stored in the granules until they need to be released. When you eat, glucose from food is broken down and absorbed into your bloodstream. Glucose then enters beta cells via glucose transporter GLUT2 and ATP is produced. The presence of ATP inhibits the efflux of potassium ions, causing depolarization. This activates calcium channels within the cell membrane, and calcium ions flow into the cell. This increased calcium concentration within the cell is the primary signal for insulin secretion. Insulin granules are released outside the cell, and insulin enters the bloodstream. Insulin then binds to insulin receptors on cells throughout the body, activating a second messenger system that causes the translocation of glucose transporter GLUT4 into the cell membranes, allowing glucose to enter these cells.